So when we stopped last time, we were talking about transcription, especially differences between prokaryotes and eukaryotes in transcription. We left off with this example that I wanted, hopefully, some of you to try over the weekend. What's the concept that we're talking about here? What part of transcription does this example deal with? Which of the steps? It says up there, sort of. Splicing. So does that mean we're talking about a prokaryotic or a eukaryotic example in this case? Okay. Prokaryotes don't splice. Eukaryotes splice. What are the black boxes? So the black boxes are exons. So the exons are numbered, 1, 2, and 3. What's the definition of an intron?
Non-necessary DNA.
Unnecessary DNA? How do we know where the introns are?
They're between exons.
They're between the exons. Okay, so how do we define the exon? Uh-oh, chicken and the egg. So what are the sequences that are in introns that let us know where the intron boundaries are? There are specific nucleotides that are at the start and the stop of every intron. There are A's, U's, and G's involved, but not necessarily in that order. So this is one of the motifs that you should remember, one of the DNA sequences that you should remember. They all start with GU and end in AG. So there will be a GU and an AG in that intron. And this next intron should start with a GU and end in an AG. There's also an A in between them that's critical to have for splicing to work. So how many introns do we have? Intron 1, intron 2. We define the introns by what's not left over in the mature messenger RNA. That is, after all of those steps of processing this primary transcript, whatever's taken out we know was an intron. So the cell defines introns for us. Whatever gets taken out by the splicing machinery is by definition an intron. So how many different splice variants, how many different ways could this primary transcript be spliced? How many different types of messenger RNAs can be produced from this primary transcript? Are you allowed to cut out exons? Does the cell cut out exons? Sometimes. So yeah, you're allowed to. What's the easiest, what's the most obvious product that would be produced from this primary transcript after splicing? Just stick the 3 boxes together. So the 3 exons rather, 1, 2, and 3. So introns 1 and 2 have been removed by splicing. We're left with the 3 exons. Okay. What else could happen? It's not just what else could happen, what else will happen in the cell. So splicing does not always happen the same way for every transcript. So let's say you have another copy of this messenger RNA, the primary transcript from this gene, second molecule of it. How could it be spliced?
Just 1 and 2.
So just exons 1 and 2? Yep. So you could have exons 1 and 2. You could have exons 1 and 3 with 2 accidentally or maybe not accidentally removed. Which probably means you could also have exon 1 somehow removed and leave exons 2 and 3. What else? Pardon? Right, sometimes when splicing happens, introns get left in. So it's not always the case that every intron gets taken out. So what would that look like? So we could have a transcript that's got intron 1 retained, but intron 2 removed. There are lots and lots and lots of different ways to splice a primary transcript, and that's just one with 3 exons and 2 introns. What's going to happen to the number of possible mature messenger RNAs that are produced when we add a 4th exon, which means there's also a 3rd intron? Sometimes different exons get removed accidentally, sometimes different introns get retained. So you get huge amounts of diversity in the number of mature messenger RNAs that are produced because splicing happens. That's the main point.
Doctor,
Yeah.
is there like some computer algorithm that you use to see the maximum number, depending on the part of the messenger RNA you want to look at to see what type of versions of the protein you can produce?
I suspect there is a pretty straightforward mathematical algorithm for figuring out how many maximum splice forms there could be. The good news, but the weird news, weird but good, is that not every splicing pattern is used by a cell for any particular transcript of any particular gene. You don't usually see one of every type. Usually, somehow, and this is one of the mysteries of molecular genetics and cell biology, is how is it that sometimes a cell will always process a primary transcript in the same way? It always take out certain exons, leave in certain introns. Different cell types do it differently. And this is one example of that, potentially. So these are all cichlid fish that come from which lake? Malawi? It was either Malawi or Tanganyika. One of the 2 rift lakes in Africa, right over here, world map. It's the first time I've ever used the world map in a genetics class, right? These are all very closely related species of fish, really closely related. If you look at their genes, they're very, very, very similar in sequence, but look at how diverse their color patterns are. Different colors, different types of stripes. Some of them have vertical stripes, bars, they're called, vertical bars, horizontal stripes. And so some scientists had this hypothesis that maybe there were certain genes that were being alternatively spliced in different ways, and that diversity that can be produced by alternative splicing, the number of different proteins you could produce from differentially spliced versions of the same gene, could be responsible for all of this diversity in pigmentation, color, striping patterns, and so forth. And they thought, well, there are a lot of genes that might be involved in this. One of the genes they focused on is a gene called Hagoromo, which, as I'm told, means dress of the gods in Japanese. Please tell me if I'm wrong. But it has something to do with patterning and pigmentation, the way the fish looks. You may or may not be able to tell. Of course, this is a zebrafish. Guess why it's called a zebrafish? Yeah, because it's got stripes. And this zebrafish on top is the normal pigmentation pattern. So it's got horizontal black stripes along its ventral, or dorsal surface rather. Now this fish on the bottom is a zebrafish, just like the one on the top, but it is mutant, homozygous, for this gene. So this gene has a loss of function mutation. The Hagoromo protein isn't produced. And look what happens to the pigmentation pattern. Those nice, clean, horizontal stripes are now disrupted. You get sort of blotches, right? Not clean stripes anymore. The pigmentation pattern is messed up a bit. So that's what led these authors to think maybe this gene, the Hagoromo gene, is involved in producing diverse striping patterns in these fish, in cichlid fish. There's a lot of different types of patterns, so make a prediction. How many exons and introns would a Hagoromo gene have to have if it was going to be responsible for making all of that variety in different striping and barring patterns in cichlid fish? A lot, a few? It turns out the Hagoromo gene, and this is from this paper from 2003, has 9 different exons. 1, 2, 3, 4, 5, 6, 7, 8, 9. This gene is spread over what do you think that is? That's a 10 kilobase pair length right there. So it's maybe 50 or 60,000 base pairs long, this gene, all of its exons and all of its introns. So look at how big the exons are compared to the introns. Every one of these vertical bars is an exon. They're tiny. Look how big the introns are. Why is this shocking? Not just because I tell you it's shocking. Why is this weird? This is very weird.
You have more junk DNA than useful DNA.
Right, this gene is mostly, RNA polymerase transcribes this whole thing. 50,000, 60,000 base pairs of this gene, right? Every exon, every intron is transcribed, turned into RNA by RNA polymerase. Then what happens? Most of it gets cut out. Introns get removed. Especially this whopper of an intron. Sometimes, what's that, like maybe 30, 40,000, 3,500 base pairs of DNA? Intron, spliced out every time. What a waste. There must be something beneficial about alternative splicing. So this is an attempt to emphasize a point I tried and probably failed to make earlier, that despite the fact that this gene has almost 10 exons, how many different types of splicing patterns could there be? I don't know, we need the algorithm now. 10 exons, 9 introns, how many different ways could you accidentally remove some exons, leave others in, leave some introns in and not? There's a huge variety of splice forms that you could produce from this one gene alone. Yet the majority, when you actually take messenger RNA out of the cells of these zebrafish, these 9 different splicing patterns reflect the majority, the vast majority of the types of messenger RNAs that are produced. So it's not the case that the cell is just randomly splicing out introns, leaving introns in. For some reason, which we're not clear about at the molecular level, for some reason these 9 are the predominant 9. So it's not random. There are specific splice patterns that occur a lot in a cell, and some that occur maybe rarely or never. And these authors have categorized them into 2 different types. So what's common to the bottom 5? Has to do with this name they've given this group right here. All those 5 share one weird thing in common. They all... Right, so they all have retained intron 5 at their 3' end. So it didn't get spliced out. There's one of these 9 that's the actual full length. That is maybe what you might have predicted before I started yammering on and on about alternative splicing. You might have said, oh, we'll just take out every intron, leave all the exons. They did find that, that's number 1. Every exon is retained, every intron is taken out. That happens. It's common. But so are the other 8 types. Exons not accidentally. Exons get taken out or introns get retained. So to make that point about exons getting taken out, here's an example where exon 3 was removed. And they see that a lot.
And these alternative splicings correlate to different fish colors?
So that's what they know. And it turns out, I'm not showing that figure, but there was a correlation. So fish, there was a very weak correlation. So whether or not their hypothesis is supported, that is that the number of splice forms of this gene, does it correlate with the number of colors in the fish? Maybe was the answer. So they're still working on trying to figure out, at the molecular level, do fish that have different color patterns, different barring and striping patterns, have more of one splice variant than the other? Still, I think, work being done. So other questions about alternative splicing, cichlids, Hagoromo? Right. Critical concept being that alternative splicing produces a variety of different messenger RNAs. Not always just one type. If not, then we'll turn our attention to something that's maybe a little bit more traditional, the fruit fly. Alternative splicing plays a really important role in sex determination in fruit flies. So how do we do sex determination? The X and Y, so let's say human. If you're X and Y, you're male. If you're X and X, you're female. What is it about being XY that makes a male male? There are two genetic differences between the two sexes in humans, is the real point here. What are the two differences between the guys and the gals? It's all right here. There's 4 letters. XY, XX. What are the two ways we differ? One sex has a Y chromosome, one doesn't. So does that mean Y chromosomes make males? What's the other difference between males and females? If it's not the Y chromosome, what? Women have 2 X chromosomes, men have 1 X chromosome. So which of the two is it that causes sex in humans? Is it the number of X chromosomes that is important, or is it the presence or absence of a Y chromosome that's important? Well, should we take a vote? In humans, it's the Y chromosome. So if you get a Y chromosome, that makes you male. It's not the number of X chromosomes. The only reason I point this out, so it's the presence of the Y that makes males in humans. The reason I point this out is because it's totally different in Drosophila. It's the other way. In Drosophila, if you've got 1 X chromosome, you're male. If you've got 2 X chromosomes, you're female. So despite the fact that Drosophila and humans both have X chromosomes and Y chromosomes, and in both species the XY genotype is male, it's for totally different reasons. So here we're going to find out about how alternative splicing causes sex determination in Drosophila. Right, something that we don't do in humans. So you can kind of ignore the rest of that. Don't worry about X to autosome ratios. Right, the important thing is if you've got 1 X chromosome, you're male, 2 X chromosomes, you're female in Drosophila. This difference, this is mysterious, the details. How many X chromosomes you have initiates a cascade of alternative splicing events. So this is how it works. When you've got 2 X chromosomes, you're female. There's a gene called SXL, stands for sex lethal. Pretty kick-ass gene name. When you have 2 X chromosomes and you're female, that's down here, exon 3 gets removed from the transcript of sex lethal. That allows females to produce one version of the sex lethal protein. So that's all on the right side of the screen. So if you're female, 2 X chromosomes, you produce the full-length sex lethal protein. If you've only got 1 X chromosome, this is the mysterious part that I'm not going into the molecular details. If you only have 1 X chromosome, poor males, you make this splice version on the left side, where exon 3 gets left in. Unfortunately for the males, exon 3 has a mutation, essentially. We haven't talked about translation yet, but for those of you that really want to know, that red bar there is a stop codon. We'll talk about that more next chapter. You can think of it as a mutation. So if you're male, you've got one X chromosome, exon 3 gets left in, and that mutation means that you don't make sex-lethal protein. So already there's a difference between, at the molecular level between males and females. Females have 2 X chromosomes, males have 1. That directly influences whether or not a fruit fly produces the sex-lethal protein. Women do, men don't. So alternative splicing. Leaving an exon that's got a mutation has an effect on the protein. What does sex-lethal do? Oh, take a guess. What process do you think sex-lethal is involved in? I'll give you a hint. We've been talking about it all day already. Splicing. So the protein sex-lethal, remember, only produced in females. Hey, it's a messenger RNA or it's an RNA binding protein. It binds to introns. So this protein is involved in alternative splicing, sex-lethal. So what does it do in this case? If you're female, here on the right side, where does sex-lethal bind? What does it look like it's, where does it look like it's binding? Right here. Is it exon or intron? It's an intron, the skinny little line connecting exons. What nucleotides is sex-lethal covering up? The 3' splice site. Remember, every intron has a G and a U at the 5' end and an A and a G. Sex-lethal is bound to that 3' splice site. It's covering that AG. Remember, only in females. So how does splicing work if you're female and you've got the sex-lethal protein? Does this exon get removed? If there's no 3' splice site, the AG can't be accessed to do the splicing. So what happens instead? Uses what's called a cryptic splice site. Cryptic just means it's not normally used. But when sex-lethal is covering up the normal AG, what's a cell to do? You go find another AG somewhere nearby and splice to that. So in the end, if you're female, you get exon 1 and then the latter half, or the 3' half of exon 2 spliced together. Because sex-lethal is covering up the place where splicing normally would have happened at the end of intron 1. So what happens in guys? No sex-lethal protein. So what's the splicing pattern going to look like? Exon 1 gets spliced out? Or sorry, yes, thank you. Intron 1 gets spliced out. And oh look, poor guys. What's wrong with exon 2?
Stop code.
Another stop codon, another mutation. Boy, to be a lady, really. So, right, splicing. Sex-lethal gets produced in females. It doesn't get produced in males because males have a premature stop codon, a mutation in one of the exons. That exon gets removed in females to produce sex-lethal. Sex-lethal prevents the retention of part of exon 2 that has that stop codon. So in females, they make normal transformer protein, TRA. Getting the picture? There's always something wrong with exons in males. Women always tend to remove that crappy part of the exon and produce normal full-length proteins. Sex-lethal, then, in this case, transformer, TRA. Guess what process TRA is involved in? Nobody? More splicing. TRA is also involved in splicing. So if you're female and you've got that TRA protein that's been produced. Now here's something we haven't talked about before. What does TRA do? It's still an mRNA binding protein. Here it is. It binds to exon 4, but in this case, binding happens in the exon. And TRA is important to make sure, if you can read that, TRA makes sure that that exon is retained, that that exon isn't accidentally removed by splicing. So when TRA binds to exon 4, the product that's the mature messenger RNA has exon 4 in it by virtue of TRA having bound to that exon. If you're a male and you never had double-sex protein in the first, TRA protein in the first place rather, your exon 4 gets spliced out. This is the end of the story. The point of all of this, the 3 steps, sex-lethal, transformer, and double-sex protein, which is what's produced by the double-sex gene here, is that finally we get 2 normal functional proteins made, double-sex that's male version on the left and a double-sex that's female on the right. Right? One of them has the amino acids encoded by exon 5. That makes it male-specific. And the other splice form, the one that's made in females, doesn't have exon 5, it's got exon 4 instead. And then those 2 proteins, male double-sex and female double-sex, go on and are actually involved in producing the actual differences between males and females. Differences in morphology, physiology, behavior, genitalia, all that. So the initial process of sex determination in Drosophila starts with how many X chromosomes you have, but then it works through this series of alternative splicing events that establishes an actual protein difference between males and females. It's alternative splicing in action. Questions, concerns, criticisms, feedback? So again, the point being alternative splicing is important, and it happens a lot, more than you'd probably think. So if on the final exam I ask, why do you think splicing is so great? What would a good answer be? Diversity is good. 3 simple words. What about these 4 different taxa, species? We've got bacteria, yeast, fruit flies, and worms. I took us off of the graph, make it a little bit less personal. What's pretty similar between these 4 species on this graph? What's on the x-axis? Number of genes. So that's different between all of these species. Bacteria, those lowly little bacteria, not so many genes. What about worms? Large number of genes. What's not different about these species? Genome size. So tell me, how is it that with a tiny little genome, E. coli has so few genes, and in worms, the genome's not that much bigger, but worms are so, and Drosophila too, let's not discount the fruit fly, right? We've got what we might incorrectly, for a biologist, consider simple organisms, complex organisms, multicellular, right? E. coli, yeast, single cell, not so complex. Complex organisms that have advanced behaviors all have about the same genome sizes. number of genes goes up. Where do you think we sit on this plot? We have barely more genes than worms. Sorry. We're somewhere here on this line. Our genome size is actually kind of up here. So we're up there in the upper corner of the plot. But we've got about the same number of genes as worms. Our genome's much bigger. It's bloated with all that junk DNA that we've talked about. Maybe bloated with a little bit of candy from Halloween too. Why are we so much more complex than worms? How is that possible given that we have about the same number of genes? Could I make it any more simple? Thank you. Splicing. Way, way, way more human genes are alternatively spliced than in fruit flies or worms. Again, what's the point? Alternative splicing vastly increases the number of types of proteins, the complexity, the diversity that an organism has available to it given its genome size. So we have a genome size that's, well, it happens to be way bigger, but it's more because we have the ability to alternatively splice more of our genes that allows us to be more advanced, colloquially speaking, not that I actually believe that, than flies and worms. We're bigger. Who knows if that's important? So that's the last step of processing. After this, we talked about capping last class, just finished talking about splicing. The third step in processing is capping, splicing, the addition of a 3' tail. And that's involved in termination, which is why we cover it last. So RNA polymerase transcribes a gene, it's cruising along the template strand, reading the DNA, producing messenger RNA, the 5' end is hanging out there in the cytoplasm, the nucleus. And then all of a sudden, RNA polymerase comes across, in prokaryotes, a particular sequence, down here at the bottom, that causes that messenger RNA. Remember, RNA is single-stranded. It likes to base pair to itself when it can. The end of the gene, there's a sequence that causes this hairpin to form, a short stretch of double-stranded RNA that forms hairpin-like shape. The formation of that shape causes transcription to terminate in bacteria. It does that essentially by that hairpin folding back and interacting with the RNA polymerase molecule itself. It's kind of like a lock and key mechanism. That hairpin, kind of like lock picking. Hairpin is used to physically release RNA polymerase from the template strand. That's what your chapter talks about as row-independent termination. That's this. There's a particular DNA sequence at the end of the gene, causes a hairpin, the hairpin causes transcription to terminate. That's prokaryotes. Similar in eukaryotes. No hairpin though, but there's still a DNA sequence at the end of the transcript that's important for causing RNA polymerase to stop transcribing. So there are two things that happen at the end of eukaryotic transcription. Both termination and the poly-adenylation, the poly-A tail, get added essentially at the same time. So two things happen. There's a specific sequence. This is another one that you should remember. So one being the splice sites, GU, and then at the other end, AG, and this one, AAU, AAA. That is a nucleotide sequence that's present at the end of every transcript. It's called the poly-adenylation signal, as it says there. And what happens is, there are a number of proteins that are hanging out physically associated with RNA polymerase as it's doing its transcription. And those are these little balls down here, these extra proteins. One of them is reading the messenger RNA molecule, essentially looking for that AAU, AAA. When that protein recognizes that sequence, that causes a protein that's closer to the 3' end to physically cut the RNA molecule. So it's like you're unrolling a roll of toilet paper, and all of a sudden one of the proteins realizes that there's an AAU, AAA written on it and rips the toilet paper roll right there. RNA polymerase is still on the template strand, but that transcript has been cut. As it says here, about 20 nucleotides downstream. So there's an AAU, AAA. And then there's about 20 nucleotides. And then it's 3' end. So what those 20 nucleotides are is dependent on the template, of course, the DNA that is being transcribed. But that AAU, AAA is always there. Then another one of these proteins does something to modify the 3' end of this transcript. That's the poly-A tail. There's an enzyme called poly-A polymerase, and its job is to add poly, lots of A's, to the end of the transcript. So what does the 3' end of a transcript look like, to summarize? When you're looking at a processed messenger RNA, what's at the 5' end? The left end in this case. That's the first step in processing. So there's an AAU, AAA here somewhere. But let's see, 5' end. This is where all the exons are. There's a cap. What nucleotide was the cap made out of? 7-methylguanosine, 7-mg, cap. So that's stuck on the 5' end. Then you've got exons, introns, depending on how alternative splicing works. Then you get AAU, AAA, 10 to 20 nucleotides, and then a long string of A's, and that's it. That's the end of the transcript. So you've got a cap. Splicing happens in between. Exons, introns get removed or not. Then the AAU, AAA, short stretch of nucleotides, and then lots of A's. What's up with the lots of A's? Oakland fans? What does the cap do to the messenger RNA again? What's the role of having that one nucleotide stuck on the 5' end? There were a couple we talked about last time, one of which is that the nucleus recognizes it, means that it's a messenger RNA that should be taken out of the nucleus into the cytoplasm. It protects the messenger RNA from being degraded from the 5' end. What does the poly-A tail do for the messenger RNA?
Is it short?
Pardon?
Does it have to do with prevention?
It does also have to do with protection of this messenger RNA. So it turns out the length of your poly-A tail dictates how long you are a functional messenger RNA in a cell. So there are also enzymes that chew up messenger RNAs from the 3' end. The longer your tail is of A's, the more time you have for those enzymes to start degrading you from your 3' end before they start chewing into the actual meat and potatoes, that is, the exons. Right? So the longer, it's very much like telomeres, which we haven't talked about much in class, but it's the same concept. That our cells add on this random, essentially, DNA sequence to the ends of our chromosomes to prevent our chromosomes from getting degraded. The same thing is essentially true of the poly-A tail. It's extra nucleotides that are added on by this one enzyme, poly-A polymerase, so that over time, yes, as translation occurs to this messenger RNA molecule, these enzymes do slowly degrade the messenger RNA from the 3' end, but it takes them a while to eat through all of those A's before they start degrading the actual exon content of the transcript. So 2 of the 3 steps in processing basically are involved in identifying this transcript, this messenger RNA, as a processed messenger RNA, the cap and the tail. The other 2 effects are they protect the cap and the tail, that is, protect the messenger RNA from being rapidly degraded. Because what's the point of going through all the work of transcribing all those introns that then got taken out, only to have your transcript destroyed by the cell? Doesn't seem very efficient. Capping, splicing, poly-adenylation, tailing, all of which are eukaryotes only. All of processing has to do with DNA sequences. We've got sequences and also transcription. We've got sequences in the promoter that proteins recognize to start transcription. We've got DNA sequences that I've asked you to remember, which you're already starting to, I can tell, that are involved in splicing. And there are DNA sequences that are involved in polyadenylation. And those are scattered throughout the gene. So at this point, when you download, or if you've already downloaded this PDF exercise from Blackboard, you should be able to do questions 1 through 6 already, and I suggest that you work on those. The one exception is there's one part of question 1 that you probably aren't going to be able to do until we talk about translation. You may know this already, but I'm asking you to label 5' and 3' untranslated regions. We'll talk about that next chapter. So you don't have to be able to do that specific part of question 1 yet. But otherwise, questions 1 through 6, you should be able to get through. Try this before next class, bring questions. Do you have issues identifying some of the parts of a gene, and so forth. Got one more tiny little thing to talk about. Application of mutation. So mutations happen all the time. Poor Drosophila have those mutations in their exons that make males, and if you're splicing out those exons, they become female. Make some predictions for me. What happens to transcription if you have a base pair change, say, in the promoter? What effect would that have on transcription? Remember, the prokaryotes have -10 and -35 consensus sequences, specific DNA sequences that which protein binds to? Prokaryotes, -10, -35. Those are called sigma factor. And what binds the Tada box in eukaryotes? It's a pretty cleverly named protein, Tada binding protein. What happens if the promoter sequence gets mutated? If those DNA sequences change because of mutation, will sigma or TBP still bind to them? They're looking for specific DNA sequences. So if mutation happens, what happens to transcription? It doesn't happen, because what do those proteins do, sigma and TBP? They bind the promoter, and their next step is what else do the proteins do besides bind to the DNA? They grab on, they bind to the DNA, and then they grab onto RNA polymerase and say, here's a gene, come transcribe it. Yeah. So transcription won't start if sigma or TBP don't bind to the promoter, because nothing brings RNA polymerase to start transcription. Okay, easier example. What happens if you mutated the G or the U or the A and the G in an intron? Can splicing happen? It could, but probably not using one of those sites. So let's say that was the sequence of this RNA molecule. Let's say mutation caused that A to become a C. So now it's GU something A and then CG. How would splicing work now? We've got the G and the U, the 5' splice site. That might get spliced to the next AG, maybe. Causes a change in splicing. And we'll see next chapter, change in splicing causes change in the protein sequence, potentially. So you get a different protein produced. Next chapter, we'll talk a lot about what happens when mutations happen in exons. I said gene, but really I should have said exon. Because all of these components, hopefully you realize at this point, are part of a gene. The promoter, the exons, the introns. We're already talking about what mutations happen in genes. What about the terminator? I promised I wasn't going to do an Arnold Schwarzenegger impersonation. I still might get through it without doing it. What happens if the AAT-AAA gets mutated? You know the answer. You just have to say it. If that's responsible for termination, then if you don't have let's say it's AGT, oh man, AGT, AAA, how does that affect transcription? Yeah, RNA polymerase up, didn't see the poly-adenylation sequence yet, just keeps on transcribing. Of course, by that point, it's transcribing what's no longer gene, because it's missed what used to be the 3' end of the gene. So what's RNA polymerase doing now? It's transcribing intergenic DNA, stuff that's not exon. It's going to make some crazy wild protein that does weird things. So mutations can have a lot of effects on transcription. And when we come back next time, we'll start talking about translation.